Hi guys, I'm sorry I can't be in class today. I got called out to do a little bit of extra training for a software program, so I can't be there. But because your test is on Tuesday of next week, I can't miss a lecture. So you're going to get one this way instead. Just be like you're sitting at home, but I still got to give you a lecture. This very last lecture will be all about the flip side of diffusion, osmosis, and facilitated diffusion. Now I'm going to start talking about all of the different transportation methods that require energy from the cell. With diffusion, osmosis, and facilitated diffusion, all of those things were diffusion. Molecules were able to flow down their concentration gradients, go from an area of high concentration to an area of low concentration, just very simply. Remember the sugar cube, how at first it's all nice and clumped together, but eventually because molecules, water and the sugar molecules, they are colliding into each other and they never stop moving, they start to spread out. And that's simply what diffusion is. Whether it's simple diffusion and I just have potassium going across my cell membrane, it's osmosis where I have water moving across a membrane, or I have facilitated diffusion where I have something like glucose going through a protein channel to get through the cell membrane to the inside of the cell. The flip side of all of that is now active transport. It's when molecules move against their concentration gradients. No longer are they moving from high down to low, they are now going the opposite way. They are going from low to high. They're going against the flow. Think about it like an ocean. Passive transport is being carried away by the current. Be a scary prospect, but that's an example of it. You're just carried along. You don't have to exert any energy in order to just move with the tide. If I wanted to get out of the current, because currents are dangerous, I would have to exert energy to get out of it. That's the same thing as active transport. No longer am I moving with the current, I am now trying to move against the current, and I'm going to have to use energy. So this type also requires energy input from the cell. And this energy is going to be in the form of ATP. This is your energy currency. Adenine triphosphate. That is the energy currency of any cell that you could ever talk about. It's a very, very important molecule, but that's how it's going to be input. One type of active transport are called ion pumps. Ion pumps move small molecules through protein pumps located in the cell membrane. If you remember back to facilitated diffusion, in facilitated diffusion, I used protein channels. Channels do a specific job. They kind of act like a tunnel where you pass under a mountain and get to the other side. Easy smeegee. Protein pumps, on the other hand, take a little bit more effort, and they do require that energy input. So protein pumps, just another protein that's located in my cell membrane, but it has a specific job. And because of that, I call it a pump instead of a channel or a pore or a receptor. It's called a pump for a reason, because it basically will take some things and then pump them out to the other side. An example of this is the sodium-potassium pump. This pump is very important in muscles and nerves. This is how we propagate signals. This is how I flex my muscles and do curls. It's how I send signals from my fingertips up to my brain. These two things are very, very important. The way that a sodium-potassium pump works is that sodium binds to the protein pump. It'll have specific spots where it will receive whatever it needs to carry. So it binds to that protein pump, which causes the protein pump to change shape. And this is what allows it to take molecules inside or outside of the cell, whatever it's being asked to do. Once it changes shape, or the protein pump transports 3 sodiums out of the cell. Now, sodium is what is going against the flow, against the concentration gradient. So that is why I have to bring in this pump. Once the 3 sodiums get to the outside, it allows for potassium to bind, causing the protein pump to change shape again, kind of revert back to where it was before, which allows 2 potassium ions to be transported into the cell. Another type of active transport is called endocytosis. Endo means in. Cyto means cell. So in cell, I am taking something in. So this is when materials are brought into the cell. How this works: a pocket forms off the cell membrane that will pinch close and carry the materials inside. What this looks like: say I have some kind of cell right here, and I have some kind of nutrient on the outside. What will happen is your cell membrane will start to make a groove, and it will take in that particle, that material, the ions, whatever it is that it needs to take. And it will create a vesicle, you guys have heard that word before, in order to take this material inside. So that eventually you end up with a whole cell again, maybe lost just a little bit of the cell membrane, but don't worry, it comes back and it'll get recycled. And it's carrying that material that it tried to take in. I can talk about endocytosis in 2 different ways, and it totally depends on what the material is that I am taking into my cell. If I am taking in food, I call it phagocytosis. This is when cells are eating. It is called cell eating. Once the vesicle is formed, this food vesicle is formed, it will fuse with a lysosome to break down the food in order to make it usable for the cell. So that's one type. Still formed the exact same way, doesn't change. It's just when I take in food that the cell can eat, and I call it something specific, and that is phagocytosis. The flip side of that, in order for you to survive, you also have to take in fluids. Same thing for a cell. When I take in fluids, I call it penocytosis. This is cell drinking. These, once they're taken inside, the vesicles that form become known as vacuoles. They become storage containers. You don't need to fuse with the lysosome, because with cell drinking, you're not taking any food in, you're just taking in the fluid. So you don't need to break it down into usable bits. Now, of course, with everything, since I talked about diffusion, osmosis, and facilitated diffusion, which were all passive transport, there also has to be an opposite for phago and penocytosis, which are both endocytosis. So my last thing that I need to talk about is exocytosis. This is when materials are exported out of the cell. And it works in the exact opposite way. I start with my cell, and I've got a vesicle on the inside that's got something in it. It's a vesicle from, say, the Golgi apparatus that has newly folded and ready to go proteins. And that vesicle will move up to the edge of the cell. The vesicle will fuse with my cell membrane, causing my cell membrane to make a little indent that will then let out everything that little vesicle was holding. If you notice, a little bit of my cell membrane got taken away when I made a endocytosis vesicle. This is how I recycle it. When I make an exocytosis vesicle, then my cell membrane parts that I used for endocytosis will come back and go back into where they need to go. So there is a recycling. That's it. Whatever time you guys have left, please work on your study guide because your test over this unit is on Tuesday. No ands, ifs, or buts about it. It will be on Tuesday. Also, if you are wanting to do the extra credit where you make me a animal cell, plant cell, or bacterial cell out of cake, cookie, Rice Krispie, whatever it is you want to make, that is due by Thursday of next week. I will accept them as early as Tuesday, but not before that. So either give them to me on Tuesday or you got to bring them to class on Thursday. Those are the only 2 days that I will accept them. Otherwise, then you just missed out on a little bit of extra credit. If you have any questions, make sure you ask me. Come find me on Monday or on Tuesday before your test so I can clear up any questions. Or you can ask me through Edmodo. I will see you guys next week.